Hello students. Now let us continue with the biofertilizers. In my last video lecture, we had studied about the production of rhizobium biofertilizer. Today let us study about Azotobacter as biofertilizer. So here we are going to study about Azotobacter, the isolation methods of Azotobacter, mass production and application of this Azotobacter in detail. So, what are these Azotobacter? As discussed previously, this is one of a bacteria which has an ability of fixing the atmospheric nitrogen, and they are free living microorganism. They are free living bacteria. They have an ability of fixing the atmospheric nitrogen. This Azotobacter is one of a aerobic gram-negative bacteria which has a slimy layer. That is, they have a thick capsule made up of polysaccharide which is been surrounding on the cell wall of a bacteria. This is one of the characteristic feature of Azotobacter. The main purpose of having this slime layer also we have studied in my previous class. That is, this slime layer helps in the nitrogenase activity. It avoids the contact of oxygen, that is, the penetration or the entry of oxygen is limited because of the presence of this slime layer. So this is an important characteristic feature of Azotobacter, and this Azotobacter, the species of Azotobacter, especially Azotobacter chlorochrocum, has been found dominant in most of the agricultural soil. Wherever the agricultural practice is happening, we could see the Azotobacter chlorochrocum in abundant, and this bacteria has an ability of fixing the atmospheric nitrogen. Say for example, 2 to 15 milligrams of nitrogen has been fixed per gram of the carbon source in the cultural media. So this Azotobacter chlorochrocum is one of the dominant group of organism which is being found inhabited in arable soils, that is the cultivated soil, that is agriculturally or active or the rhizosphere soil. It is found in dominant, and this is used as a biofertilizer for all non-leguminous plants. So we know leguminous plants, rhizobium is one of a organism which can invade and fix the atmospheric nitrogen. If the plants are non-leguminous, better to go for Azotobacter biofertilizers. Say for example, rice, cotton, vegetables or any fruits producing plants. For all such plants, we can go for the biofertilizer, the preparation of the biofertilizer made up of Azotobacter. Azotobacter cells are not present on the rhizoplane, but they are abundant in rhizosphere region. Rhizosphere region is a region where you could find the activity or the predominantly this Azotobacter is being found rather than that of a rhizoplane. So you know, rhizoplane is a surface of the root surface, the soil or the microorganism which has been found on the surface or just beneath the epidermis of the root. Those microorganisms are called as rhizoplane microorganism. Rhizosphere, the organism which is, or the soil which is surrounding the root system, and the microorganism which is present in such soil is called as rhizosphere soil. So this Azotobacter is one of a bacteria which is predominantly found in rhizosphere soil. Coming to the isolation of Azotobacter from soil. So the source of isolation is the soil, whereas for rhizobium the source was the root nodules of leguminous plants. However, for Azotobacter, since it is one of a soil microorganism, free living microorganism predominantly found in the soil, that is the reason the soil is one of a source which we can use to isolate this Azotobacter, especially the active soil where the microorganism activity is more. That is, the rhizosphere soil is the soil which we can use to isolate such azotobacter. There are a number of media which we can use, out of which one of the best media which we can go for is Waxman number 77. Likewise, we can use nitrogen free ginseng media, ash based media, azotobacter media. Many media are there. I have given the composition of Waxman medium number 77. So the main ingredient in this particular media is the mannitol. Mannitol is one of the important carbon source which this bacteria has an ability of utilizing this mannitol. And the calcium carbonate is also one of the source which is being found useful for the isolation of this azotobacter, and also it forms helps in the formation of the slimy layer around the each of the bacterial cells. And this Waxman number 77 or ash base or Azotobacter media is one of a specific media. It's a specific media for the growth of only the Azotobacter. So it's a specific media or the special media for the growth of Azotobacter. So that we have to remember. Once again, the sample which we are using is the rhizosphere soil, fertile soil, and the media which we can go for is nitrogen free ginseng media or Waxman number 77 media, or an azotobacter media, ash based media, etc. So this media is has to be prepared and sterilized and kept ready. So since we are isolating this azotobacter from the soil, we can go for the technique which we can follow is a serial dilution method. As we all know very well about the serial dilution method, 10 grams of rhizosphere soil is mixed well in 100 ml of sterile distilled water, and the suspension can be serially diluted, and it can be plated out onto the specific media, as I had mentioned earlier, Jensen media or Waxman number 77 or ash base or azotobacter media. Better to go for pour plate method or plate method or pour plate method. Even streaking can also be done. Since we are serially diluting, if you want to enumerate the number of azotobacter present in the soil and to get the isolated colony, better to go for, because the purpose of going for serial dilution, spread plate and pour plate, to get the isolated colony, to get the different species of azotobacter. As we all know, this is one of the media where only Azotobacter can grow, but there are many species of Azotobacter. So in order to distinguish them and also to get the isolated colonies of each species, better to go for spread plate or pour plate method. Always better, spread plate is better because the temperature of the agar may also matter. Even we can go for a pour plate technique also. So any of these method can be followed for the isolation of the azotobacter. So once it has been inoculated onto the media, and the incubation temperature is 28 to 32 degrees Celsius. 30 degrees Celsius is the optimum temperature for the growth of these soil bacteria. So incubation has to be done at this range of temperature for around 24 to 48 hours or 3 days. Once the incubation period is over, once we observe the colony, Azotobacter grows as a soft, milky, mucoid colony on the surface of the media. So, once the colony grows, the important characteristic feature is formation of soft, milky, mucoid colonies. So that is the characteristic feature of this Azotobacter. So once it has been grown, it can be transferred into the liquid ginseng media or ash based media, whichever the media is being used for the isolation, can be transferred and kept in rotary shaker for few days. This is for the increasing the amount of inoculum. So that gives the starter culture for the mass production. So the colonies of Azotobacter on ash base or Waxman number or Jensen media, you could see in the image, you could see milky, translucent, they are translucent mucoid colonies. These are the important characteristic feature to see whether it's an Azotobacter. However, the media is specific, only Azotobacter could grow on these media. So once again I will repeat, the source of inoculum is soil, and the media which we can use can be of any media, specific media for Azotobacter. Say for example, Waxman number 77, Jensen media or ash base media or Azotobacter media. The carbon source which is required for the growth of this organism is mannitol, and the calcium carbonate source is important for the growth of this organism. Soil sample, rhizosphere soil sample has to be brought, serially diluted and plated out onto the media. Any method can be followed, either a spread plate or a pour plate method can be followed. Incubation has to be done for 2 to 3 days at 28 to 32 degrees Celsius. 30 degrees Celsius is the optimum temperature for the growth of this organism. After the period of incubation, you could see observe the colonies, which are soft, milky, mucoid colonies can be observed on the surface of the plate. Once you get those colonies, you can subculture onto the slants, or any other preservation methods can be followed. Either you can go for lyophilization or subculturing, or the transferring of the inoculum from one media to a fresh media frequently. That's called as subculturing. You can store at 4 degrees Celsius, or you can overlay the mineral oil, or you can also preserve them in paraffin. So these method can be followed, and you can call it as a mother culture. So using this mother culture, you can have any such culture. One mother culture has to be kept separately, and you can go for the further identification. Biochemical test has to be, which I'll be talking in my next slide. For knowing which species it is, we can go for further biochemical tests, or you can follow the new techniques. Any other molecular techniques can be followed for identification of this organism till species level, either by DNA sequencing or any other methods can be followed. So once you obtain this mother culture, in order to prepare a starter culture, transfer a loop full of inoculum first into the 50 ml of the media, and keep it in shaking incubator for some 24 to 48 hours. You could see the growth of this organism that can be used as a starter culture. So, how to identify this Azotobacter? As I was mentioning, the identification can be done by the traditional method, that is following the Burgess manual, that is by performing various biochemical tests, or by going for the molecular characterization. So here I have given some of the biochemical tests which shows positive or negative test for Azotobacter. So they are gram negative rods. They are gram negative rods. They are capsulated. They are aerobic. They are aerobic, and they have PHB granules inside them. These are some of the microscopic characteristic feature. Once again I will repeat. They are gram negative bacteria. Gram negative rods. They are gram negative rods, and they are aerobic. They are capsulated and have PHB granules. You have studied all this, how the staining of techniques of all this you would have studied in your laboratories. You know about that. And also these Azotobacter, as the culture become old, they have an ability of producing various pigments, melanin pigments. That also I am going to tell you in my next slide. They produce pigment, and also they have an ability of becoming a cyst, that is they lose their morphology. They become slightly coccoid, that is round, and they secrete a thick layer around it. Those are called as cyst. That is also an important characteristic feature of this Azotobacter. And the biochemical tests, they are indole positive, citrate positive. You can see the images in this particular slide. Indole positive, and they are MR positive, VP negative, and glucose fermentation they are found to be positive. Starch hydrolysis, they will hydrolyze the starch. And another thing is they are mannitol also positive, catalase positive. These are some of the biochemical tests of Azotobacter. However, by isolating them on the ash base media or on the Azotobacter media or Waxman number 77, etc., since it's a specific or a special media, hence the growth of on such media itself suggests that they are Azotobacter. And also we can go for this biochemical test, or we can go for any of the molecular techniques for identification these organisms till species level. That is more important. As I was mentioning, Azotobacter, the characteristic feature of this Azotobacter is also the production of the melanin pigment. Melanin pigment production happen as the culture become old. That is important, which we have to notice, and also would have observed this in our laboratories also. The colonies which become black or it produce brownish pigment once the colony become little old. This is due to the production of melanin, which is due to the oxidation of tyrosine by producing a tyrosinase enzyme, and the color can be noted even on liquid forms as well as on the colonies. Some of the pigmentation has been described. So different species of Azotobacter has an ability of producing various kind of pigments. Say for example, Azotobacter corococcum produces brown blackish pigmentation in liquid medium. Even on liquid medium, the images over here, even on solid medium also, they have an ability of producing. On liquid medium especially, they produces brown black pigmentation. Azotobacter bigerenchi, they produces yellow light brown pigmentation in liquid media. Likewise, Azotobacter weinlandi, they produces green fluorescent pigmentation. Azotobacter paspali produces green fluorescent pigmentation in liquid medium. Likewise, Azotobacter macrocytogens produces pink color pigmentation. If the pink color pigmentation is being produced, that is one of the characteristic feature of Azotobacter macrocytogens. That indicates it's Azotobacter macrocytogens. That is how we can identify them. Azotobacter incygens, they produces grayish blue pigmentation in liquid media, and green fluorescent pigmentation is also being produced by Azotobacter agilis. These are some of the pigmentation, the melanin pigment they produce that varies from one species to another species. Another important thing which we have to note down for Azotobacter is Azotobacter, apart from having the ability of fixing the atmospheric nitrogen, they also have an ability of producing siderophores. Siderophores are the substance which has an ability of showing the antagonistic characteristic feature. The organism which produces this siderophores, like for example Pseudomonas fluorescens, it produces a siderophores which is an iron chelating agent. That is, they removes the iron from that particular region so that that becomes the antagonistic activity for other pathogens. So this apart from that particular character, this Azotobacter is also one of a PGPR. You know what are PGPR? That is plant growth promoting rhizobacteria. They have an ability of producing various growth promoters like auxins, gibberellins, etc. This, because of having this property, apart from nitrogen fixation, antagonistic property, as well as PGPR activity they have, hence this Azotobacter has got a highest importance as a biofertilizer. So now coming to the mass production, as we had studied it for rhizobium, the method or the technology is the same. So once you isolate and identify the organism, you will prepare the mother culture. From mother culture, we will preserve the mother culture, and one of the culture can be used for the preparation of starter culture. That is scaling up of the amount of the inoculum. So that is loop full of inoculum, inoculate into the 50 ml of the media. This 50 ml of the inoculum, once it the organism grows, you can scale up to 500, then into 1000 ml of the media is enough for around 100 liters of the broth which we are using for mass cultivation in the fermenter. So once the starter culture is ready, keep the fermenter also ready. Add the sterile media. Which media? Either any of the broth, Waxman number 77 or ash base or Jensen media. Any of these media, sterile media is being added into the sterile fermenter. The temperature, optimum temperature 30 degrees Celsius is required, and also the pH has to be maintained to 7. And proper aeration, since it's an aerobic organism, proper aeration has to be done, proper agitation has to be done, and leave it for the growth for around 5 to 6 days. At that particular duration, the condition which is required for the growth has to be checked properly. QC, quality control, has to be done by taking out some amount of the growing organism and check for any contamination, and at the same time the number of cells at the stationary phase, around 10 to the power of 8 cells per ml. 1 ml of the broth should have approximately 1 crore of the cells should be present in 1 ml. If that reaches, if the cell count reaches to 10 to the power of 8 cells per ml, that is the condition which is required according to Indian standard. So it indicates that the fermentation or the growth phase is done. And at the same time, we can go for carrier base. So the carrier base here also, either peat lignite or charcoal can be used. So prepare this peat lignite or charcoal, make it into fine powder, sterilize and neutralize using calcium carbonate. The neutralization, that is the pH which is required, is 7. The finely powdered neutralized carrier base has to be sterilized in an autoclave. Once it is sterilized and it is cooled, the produced Azotobacter is mixed with such carrier based inoculum, and it is spreaded on a thin polythene sheet, dried in shade, and it has to be packed in a polythene bag, and it can be stored at 4 degree Celsius. This is for carrier base. If it is a liquid base, it is need not have to go for any such carrier base, solid carrier base. The broth itself is enough for the process of dispatching in a bottles. The main disadvantage of using the solid carrier base is, once we are mixing and once we are spreading it on a polythene bag, the chances, polythene sheet for drying, the chances of contaminations might be there. So to avoid this, the new technology, that is liquid biofertilizers, are also available. So that will also serve the purpose. However, the liquid biofertilizer production, it's comparatively costly, but it has got various advantage. However, this carrier base, they are the cost is cost effective, but the chances of contamination might be there. There's a difference between the liquid as well as the carrier based inoculum. So that's what, from for mass production, 1 liter of starter culture to 100 liters of medium is transferred onto the bioreactor or fermenter. Once the Azotobacter attains the concentration of 10 to the power of 8, please remember it is not 108, 10 to the power of 8 cells per ml, the broth is harvested and it is ready to make a carrier based inoculant. A suitable carrier base is finely ground powder, that then it is just mixed with the carrier base, and it is cured for a week, and then it is packed in a polythene bag, and it is dispatched. So these are the biofertilizer, which you could see the image of biofertilizer once it is ready. This is how it looks. The carrier base can be either a peat, lignite, or a charcoal powder, finely ground charcoal powder. Once it is neutralized and sterilized, the produced inoculum is being transferred or mixed with this and dried, and it has to be packed in a polythene bag. Usually in the 200 grams of a biofertilizer is being found available. I will tell you later in my previous, in my next video lecture, what are the things has to be written on this polythene bag. What are, according to Indian standard, what are the how actually this polythene bag should be, and what are the how the application of these biofertilizers has to be done, what is the temperature we have to maintain to store this, all such directions has to be written on the polythene bags. So this is about the mass production of biofertilizer, that is Azotobacter. Coming to the methods of application of Azotobacter biofertilizers. How to apply applications. As we all know, seed treatment. Seed treatment, you are going to mix the seeds with the slurry of Azotobacter. So how to prepare this slurry? Slurry is nothing but the mixture of this Azotobacter with water. That's called as slurry. Some amount of water has to be taken and you mix the Azotobacter fertilizer, whatever is we have brought, that has to be mixed and made into slurry. And once it has been made, the seeds which we want to sow has to be dipped in such slurry for overnight. Later it has to be dried, taken out, and it has to be dried, and immediately it has to be used for sowing in the field. That is about seed treatment. Seedling treatment, the same kind slurry has to be prepared. If you are, there are certain plants where we have to sow the seedlings which has been brought from the nurseries. There are some crops where we are going to sow the seeds. If you are going to use seedlings, then such seedlings has to be dipped in a slurry for 10 to 15 minutes. That is, especially the roots of the seedlings has to be dipped in the slurry, and it has to be maintained for 10 to 15 minutes in such condition. Then the seedlings which have been treated has to be transplanted into the main field. That is about seedling treatment. Then pouring of slurry for crops, that is slurry which we have prepared, that can also be just poured onto the near the root zone of the crop. That is, you can dilute this with water, and the small amount of the slurry is poured near the root zone. Wherever you can find the roots of the crop, there you can add some amount of slurry. That is called as pouring of slurry for crops. Top dressing. For example, for cereal crops like wheat, jowar, rice, ragi and all, Azotobacter bioinoculant, whatever we have got from the market, that the farmers, what they can do is 2 kg of the bioinoculant is required for 1 acre. That is, 2 kg of Azotobacter inoculant can be mixed with farmyard manure. You know, farmyard manure, just a compost which has been prepared by the farmers. 20 to 25 kgs of these farmyard manures has to be mixed thoroughly with 2 kgs of Azotobacter, and then it can be broadcasted by top dressing after transplantation. After transplanting these crops, you can just sprinkle this biofertilizer which has been mixed with farmyard manure on the surface of the soil. That's called as top dressing. So these methods can be followed for using this biofertilizer onto the fields. So once again, which are those? Seed treatment, that is treating the seeds which we are going to sow by making the slurry of the biofertilizer. Seedling treatment, dipping the roots of the seedling on the into the slurry for around 10 to 15 minutes, and then it is being transplanted onto the main field, or pouring the slurry near the root zone of the crop, or sprinkling the mixture of farmyard manure with the bioinoculant of Azotobacter on the surface of the soil after the process of transplantation of the plant, or the the field where the farmer has grown their crops. There we can just sprinkle, that's called as top dressing.